Hi and welcome to the Minecrafted Protein Factory, a fully working model of protein synthesis within Minecraft, built as a part of the University of Edinburgh's Principal Teaching Award Scheme. My name is Simon Parker, and today I'll be taking you around what I've built within Minecraft, as well as outlining the scientific background of this project. Before working with the build itself, there's some theory to go through for anyone who doesn't know what protein synthesis is or how it works. For the rest of you, skip to 143, the timestamp mentioned just then. First, remember that DNA is used to generate messenger RNA or mRNA in a process called transcription. After this, mRNA is used to generate a protein in a process called translation. For transcription, we need to know what the structures of DNA and mRNA are. Each DNA nucleotide has three components: phosphate, deoxyribose, and a base. Importantly, each base could be one of four options: adenine, thymine, cytosine, or guanine. In a cell, however, DNA comes in two strands: the coding strand, which is on the bottom here, and the template strand, which is on the top. The coding strand codes for the building blocks of proteins, called amino acids. There are 20 such amino acids, which each map onto a 3-base DNA triplet codon. Note that adenine is always bound to thymine, and cytosine is always bound to guanine. Next is messenger RNA, or mRNA, which is the same as DNA apart from two features. First, all thymines are replaced with uracil. Second, deoxyribose is replaced with ribose. That's all you need to know about the structures involved with transcription. However, before we do any transcription within Minecraft, we have to build our own oligonucleotide, or short length of DNA, using the world. We're making an oligonucleotide that is 12 base pairs in length. This means it will code for 4 amino acids, namely glycine, phenylalanine, proline, and leucine. Now we can finally use our Minecraft world to generate an mRNA strand. First, the DNA strand I made is unzipped. Next, mRNA is synthesized off the template strand. In a real cell, the DNA zips back together again, but in this case, it just kind of disappears whilst the mRNA comes to rest on the plinth in front of you. Next comes translation, which occurs in 3 steps, namely initiation, elongation, and termination. Translation uses 2 cellular components you should probably be aware of the structure of, namely the ribosome and transfer RNAs. The ribosome is split into 2 subunits, a big one on the top and a smaller one on the bottom. It also has an E site, a P site, and an A site. Transfer RNAs, or tRNAs, meanwhile, are just taxis for amino acids. Each tRNA carries an amino acid on its top, along with a specific anticodon on the bottom which relates to the right sequence on the mRNA. At this point, I think it's time for us to put all this theory into action in Minecraft. Initiation occurs when an initiated tRNA, which is bound to the small subunit of the ribosome, recognizes a start codon in the mRNA. Once the start codon is recognized, the larger ribosomal subunit joins the party. Now we move on to elongation. A transfer RNA with the correct anticodon enters the ribosome in the A site. Next, the bond between the initiated tRNA and its amino acid, which is always methionine, is transferred to the amino acid of the tRNA in the A site. The 2 tRNAs then move left towards the E site, from which the empty tRNA then leaves. A new tRNA then enters the A site, and the cycle begins to repeat. All good things must come to an end, however, so once a stop codon enters the ribosome, termination begins. Instead of a tRNA, a release factor enters the A site. It cleaves the bond between the amino acid chain and the P site tRNA. The whole complex then dissociates, leaving only the polypeptide chain. In this case, Minecraft then tells you of its composition. And that is protein synthesis in Minecraft. Thank you for watching. Other Minecrafted projects are upcoming, including a model of infectious disease spread. A map file may be released now or in the future. Please contact the email address below for more information. If a map file is made available, a link will be provided in the description. Again, thank you for watching and have a wonderful day.